we have studied about viruses and the structure of virus, the characteristics of virus. Now microbes are organisms which can be seen only under microscope. They are not visible by the help of naked eye. So bacteria, viruses and even some protozoa and fungi, they are all included under microbes. So viruses, we have studied about how they undergo replication by the help of the lytic cycle, by the help of the lysogenic cycle, then structure of a T phage, structure of the RNA virus, that is an example of TMV, then economic importance of virus. So after studying virus, we shall now study about bacteria. So everything about this bacteria. Now bacteria are living cells. When we compare to virus, these are developed, these are clearly living cells. But however, they are prokaryotes. The bacteria, they are having developed cells when we compare to virus, but they are primitive cells. Among the cells, when we compare, they are primitive cells. They are the simplest organisms which are existing on this earth, and also they are the smallest type of cells which we can see. Now bacteria are prokaryotes, and according to the Whitaker's classification, 5 kingdom classification, they are classified under kingdom Monera. So bacteria are classified under kingdom Monera according to the 5 kingdom classification of Robert Whitaker. Now however, bacteria are classified into different groups by different individuals. Some of the taxonomists, they include bacteria under plant kingdom. So they include under plant kingdom. Under plant kingdom, they classify them as a separate class, a separate group. Then some classify bacteria as a separate group other than plant kingdom. So there is a kind of variation in the classification, but whatever the type of variation we come across, the structure and their aspects, related aspects, is one and the same. So bacteria, they are living cells. So bacteria were first observed by Anton von Leeuwenhoek in the year 1675. So in 1675, Leeuwenhoek, he first observed bacteria. So bacteria were seen as small cells by Leeuwenhoek. But later, though they were observed by Leeuwenhoek, the structure was not given by him about the cell structure and their other related aspects. Later, it was Louis Pasteur and Robert Koch in between 1870 and 1876. So in between 1870 and 1876, Louis Pasteur and Robert Koch, so they demonstrated that bacteria, they cause diseases in human beings and as well as animals. But they did not mention about plants. So the diseases they cause diseases in animals and also in human beings. That was demonstrated by Louis Pasteur. And Louis Pasteur studied more about bacteria and gave more about information about bacteria, and that is why he is regarded as the father of bacteriology. So that means he is the expert in bacteria, about the bacteria. Then about plant diseases. So that is about Louis Pasteur and Robert Koch. But in plant diseases, in plant also diseases are caused by bacteria. That was shown by another scientist, Burrill. So T.J. Burrill, he first showed that the bacteria also are responsible for plant diseases. That means they also cause plant diseases. That is about just a brief introduction of bacteria. Now the characteristics of bacteria. So bacteria are prokaryotes. They are having very simplest and smallest cells. The cells are very small and they are very simple. So among the plant kingdom, they are very simple. So I said bacteria are included under plant kingdom. They are considered as one of the simplest among the plant kingdom, and also they are the smallest among the plant kingdom. And these bacteria, they don't contain chlorophyll. They are devoid of chlorophyll, and automatically they are heterotrophic in nutrition. So they are mostly heterotrophic in nutrition. Either they are saprophytic or parasitic. So some of the bacteria are saprophytic and parasitic. And when we are saying that they are lacking chlorophyll, so in strict sense, some bacteria contain chlorophyll. So this is a general character. Bacteria don't contain chlorophyll. But when we observe the bacteria in detail, there are certain bacteria which conduct photosynthesis. So they contain chlorophyll, but however the chlorophyll which is present, it is different from the chlorophyll of higher plants. So here the photosynthesis is also somewhat different. So these are photosynthetic bacteria. They conduct photosynthesis. They prepare their own food material. So in general, bacteria don't contain chlorophyll. So they are heterotrophic in nutrition. Then when we come to the reproduction, usually the reproduction method is binary fission, the common method. Most of the bacteria are reproduced by binary fission. There are certain bacteria which reproduce by conjugation, that is a kind of sexual reproduction which is seen in certain bacteria only. And bacteria are unicellular organisms. They are unicellular. But though they are unicellular, they may appear filamentous or colonial. That means the cells, they become arranged in a line. Suppose it is a single cell, these cells may arrange in a line and appear to be filamentous. And sometimes the cells, they are found in groups, they are colonial in nature. They form a colony. So that is why it is called as colonial. So this is how the bacteria, they are found, they appear in the nature. They are unicellular, this is basic, but they appear to be filamentous. They occur in filamentous forms. They occur in colonial forms, means they occur in a colony. Though they appear in filamentous or colonial, but however, there is no any division of labor in this group of cells. So when they occur in a colony, each cell is independent. They do not share their duties in that group. So when there are many cells in a group, some cells will do for certain function, some cells will do another other function. So not such kind of division of labor we find in bacteria. So this cells which are found, they are usually embedded in a mucilage cover. When such filamentous or colonial forms are seen, they are found to be covered by or enveloped by the mucilage material. That is how this bacteria occur. So when the cells are found in a chain, that is this in filamentous forms or groups, but they are their own, each individual cell carries their own function, their own metabolic process, and they are already, I mentioned, they are independent of each other. They do not depend on the neighboring cell, and there is no any such kind of division of labor. So that is about the general characteristics of bacteria. So then about the cell structure of bacteria, how the cells are found. So the cell, a normal bacterial cell, it is having a cell wall. Now this cell wall is not made up of cellulose. The cell wall is made up of mucocomplex. So mucocomplex means it is a mixture of proteins, polysaccharides and lipids. So certain proteins, certain polysaccharides and certain lipids, they form a complex form, and they constitute the composition of the cell wall. So the cell wall is made up of mucocomplex, or sometimes it is also referred as mucopolypeptide or mucoprotein, like that. So overall it is a mucocomplex, we can say, because it is a mixture of proteins, polysaccharides and lipids. And this cell wall, which is made up of mucocomplex, it is rigid in nature. So because of its rigidity, it gives a particular shape to the cell, and it gives a kind of firm nature to the cell, and that is the cell. Sometimes the cell is also made up of this chitin. So usually the cell wall is also made up of chitin and also mucocomplex. And inner to this cell membrane, it is the cell membrane. Inner to this is the cell membrane, and this cell membrane has many folds like this. So these folds are present, projecting towards the cytoplasm area. So that is the cell membrane. So this is the cell membrane which we can see. Now outer to the cell wall, there may be any other one more protective layer, which is usually made up of gelatinous nature, or sometimes a type of slimy layer. So this gelatinous sheath is usually referred as capsule. So sometimes you find an additional sheath here, so that is called as the capsule. So this is present only in some bacterial cells, not all bacterial cells. Usually the pathogenic bacteria which cause disease, they are protected by this sheath, or which we call it as capsule, and it is made up of gelatin, or sometimes it is a slight slimy layer which we can find in the this cells. Then if you come to the other parts of this cell, so the cell wall, about the cell wall. So the cell wall of some bacteria is having a characteristic reaction. So this cell wall shows a characteristic reaction towards Gram's stain or Gram stain. So the cell wall in certain bacteria show a kind of characteristic nature. So Gram stain is a kind of stain. It is a chemical. So this Gram's stain, it was first observed by a scientist called as Hans Gram, a Denmark actually bacteriologist he is, a bacteriologist from Denmark. So this Danish bacteriologist Hans Gram, he actually prepared a kind of stain. He developed a kind of stain which that stain was named after him, called as Gram's stain. Now this Gram stain is a standard technique today, even which is used today, and it is used to classify bacteria into two types. So the Gram's stain, when it is treated to the cells of bacteria, certain bacteria they respond to this Gram's stain. So they retain the stain in their wall. So when Gram stain is added to the bacteria, some of the bacterial cells they retain that stain. They respond to that stain, and such are called as Gram positive bacteria. Those bacterial cells, or if the cell wall retains the Gram stain, and if it accepts the Gram stain, it is called as Gram positive bacteria. Certain bacteria will not accept that. They do not allow the stain to retain in the cell wall. So hence they are called as Gram negative bacteria. So positive means they receive, they accept that stain. The stain is retained in the cell wall, and they are called as Gram positive bacteria. In certain other bacteria, the stain is not accepted. The stain is not retained in the cell wall. In other words, the stain is rejected by the cell wall. So that is called as Gram negative bacteria. So hence the Gram stain helps in classification. So this is a standard technique which is used today to classify the bacteria, and that is called as Gram stain. This is to show the this characteristic reaction to the stain, whether it is accepted, that is retained, or it is not retained. Then usually the bacteria are also having long thread-like structure called as flagella, just like how the Liglina and all that. But not all bacteria are having flagella. So this flagellated bacteria, they are having usually a slimy layer, that is the sheath or the capsule, which is a slimy layer, and the cell membrane inner to the cell wall, it is folded inside. So these folded structures are called as mesosomes. So mesosomes are the structures which help in the formation of the cell wall, or they help in the production of energy. So like mitochondria. So these mesosomes are the folds which are produced by the cell membrane. Then in the cytoplasm there are 70 S ribosomes. The cell contains 70 S ribosomes in it. So the cell membrane produces mesosomes, and the cytoplasm contains 70 S ribosomes. So ribosomes are classified on the basis of their sedimentation coefficient, measured in the terms of Swedberg units, written as S. 70 S means 70 Swedberg units. It is the property of sedimentation which is measured in ultracentrifugation in terms of Swedberg units, and in bacteria we find 70 S type of ribosomes. However, in eukaryotes, in the higher organisms, there are about 80 S ribosomes. So these ribosomes, they are freely present in the cytoplasm. And as there is no endoplasmic reticulum here, so there is no question of the attachment of the ribosomes to the endoplasmic reticulum. So the cytoplasm also contains vacuoles in it. There are small vacuoles found in the cytoplasm. In addition to that, there are some food materials like oleutin granules and also glycogen granules are also present in the cytoplasm. Then some bacteria, especially these autotrophic bacteria. So autotrophic bacteria means they can prepare their food by their own. For example, photosynthetic bacteria containing chlorophyll, they contain chromatophores containing the pigment. Chromatophores are structures which contain the pigment, chlorophyll pigment. So there are no well-defined chloroplasts. In higher organisms, in plants, we find chloroplasts, isn't it? But here we find chromatophores. So these chromatophores contain chlorophyll and they are capable of photosynthesis. So in these autotrophic bacteria, we find chromatophores. And even in chemosynthetic bacteria, they are also under autotrophic. Means under autotrophic, we come across photosynthetic and chemotrophic. So by the oxidation of the compounds, they obtain energy and they also contain chromatophores in them which help in the concerned reactions. So the mesosomes, which are the invaginations of the cell membrane or the plasma membrane, so they help in the formation of the cell wall. So whenever this cell divides into two by binary fission, the cell wall which is newly synthesized in between the two, in between, it is actually by the help of the mesosomes, and they also help in production some extracellular substance in the cells which is necessary at the time of cell division. And then if you come to the nucleus does not contain nuclear membrane. So the nuclear membrane is absent. So this is the incipient nucleus or indefinite nucleus, which is also referred as nucleoid. So nuclear membrane is absent. There is no any nuclear membrane surrounding the nucleus. The nucleus contains large amount of DNA only. No question of chromatin or chromosomes. A definite nucleus, a nucleus which is surrounded by nuclear membrane, has chromatin, which for later form chromosomes at the time of cell division. But in indefinite nucleus or referred as incipient nucleus, there the nuclear membrane is absent and there is only pure DNA and no chromatin or chromosomes. So this DNA is present in the incipient form of nucleus. So this nucleus does not contain any nuclear membrane and it contains only fine DNA strands but no chromatin. So chromatin means the DNA is actually associated with protein. So when the DNA is associated with the nucleoprotein and present in the equal proportion, means 50 is to 50, half DNA and half protein constitutes the composition of the chromatin or chromosome, and but in this incipient nucleus there is pure DNA strands present in the indefinite nucleus. So that is about the structure of the bacterial cell. Now in addition to this, we may also have some circular DNA molecules present in the cytoplasm. So these circular DNA are called as plasmids. So this is called as a plasmid. Followed. And plasmids are not concerned with the nucleus. Plasmids are circular DNA molecules which are autonomous and they are self-replicating units. They are hereditary units. They contain genes, and these plasmids, they are freely present in the cytoplasm and they are very helpful in the study of biotechnology. So plasmids are present only in bacteria, and mesosomes are also found only in bacteria. So that is about the plasmids. Then after studying the structure of bacteria, so bacteria, they possess flagella. Flagella is a long thread-like structure. Again, the number of the flagella may vary from the species to species. So again, usually bacteria are classified into 4 types based on the shape of the bacterial cells. Now, if the bacterial cell is round, it is called as coccus bacteria. So if you take the shape of the bacteria, if it is round, it is called as coccus bacteria. Suppose if the bacteria are rod shaped like this, so it is called as bacillus bacteria. And suppose if the bacteria they are somewhat spirally, means spiral shaped bacteria like this. This is spirally bacteria. And suppose if the bacteria is somewhat comma shaped, half moon shaped like this, it is called as Vibrio bacteria. So this Vibrio bacteria is comma shaped. Usually the bacillus, the spirally and Vibrio bacteria, though they possess flagella. Again, the coccus bacteria rarely they possess flagella, but flagella, they are the parts of the cell which help in locomotion. Bacterial cells possessing flagella, they help in locomotion. They can move from one region to another region. And again, the number of the flagella differs. There may be only 1 or 2 flagella or many flagella, and again the arrangement of the flagella is different in different species. Sometimes the flagella are uniformly distributed around the cell, on the outer part of the cell. Sometimes the flagella are present in tufts or in groups, either at the 2 ends. So one group of flagella at one end, another group at another end. Like that, the flagella arrangement, number of flagella differs from species to species. That is about the structure of the bacterial cell, a typical bacterial cell. Now, if you come to the reproduction of the bacteria, so there are different methods of reproduction. So one type of reproduction method, so that means there is a asexual and sexual type of reproduction. So asexual reproduction takes place by 2 types of bacteria, 2 types which are called as the binary fission and endospore formation. So under asexual, we find binary fission or only fission, we can say. It is written as only fission, and endospore formation. These are the 2 types of methods, asexual methods, which occur in bacteria. And the sexual type of bacteria, that is seen in some species. So now fission is the common type of reproduction. So almost all bacteria, they reproduce by fission. And this asexual type of method, that is binary fission, or we can say only fission, we can also call it as binary fission. There is no any difference. Now binary fission is a simple method of cell division which occurs during favorable conditions. Here the cell simply divides into 2 cells. But however, the division of the cell is not by mitosis. So there is no any formation of spindle fibers or mitotic apparatus, so that the cell divides equally into two. So it is a simple multiplication method where the cell divides. Now initially the cell which is to undergo binary fission, it elongates slightly. Now as the cell elongates, the cell will divide into two. The incipient nucleus, the DNA material, also divides into two. And the protoplasm, that means the cytoplasm involving all the inclusions like ribosomes and all that. So it also divides into two equal halves by transverse wall or constriction. So usually by the formation of a cell wall, the cell will divide, and thus the two daughter cells which are formed, so they grow. So when the cell divides into two, this cell grows and this cell also grows to attain the size of the parent cell. That is how the cells they divide, and each cell divides in about 20 minutes. So any bacteria, whatever type of species it is, all bacterial cells, each bacterial cell divides by binary fission in the period of 20 minutes. Suppose there is one bacteria, in 20 minutes it becomes 2 cells, then in the next 4, next in the next 20 minutes 8 cells. So for every 20 minutes the number of the cells go on will become double in number, and thus increase in a favorable environment. So when the environment is favorable, so the bacterial cells they divide by fission. But so this favorable environment means not only food and water, it is also the environmental factor which is taken into account. It may be temperature, it may be humidity. So all those factors, environmental factors and nutritive factors, so they are when they are favorable, so the cells divide by binary fission, and they increase in number, they become double in number, and that is about binary fission, which is an asexual method. Now in endospore formation, when the cell is facing adverse conditions, when there is a scarcity of food, water, or when there are adverse environmental conditions, so that the cell will undergo what are called as endospores. So endospores, they are also one type of structures which we have studied previously in algae and fungi, where the cell, suppose this is a bacterial cell, now this bacterial cell will form a thick wall around it, and this is called as endospore. So endospore, it is a cell which is well protected by a thick wall, and this thick wall is highly resistant to the environmental conditions. So this cell which becomes surrounded, so endospores, this is called as an endospore. These endospores are also called as the resting spores, because these cells, they take rest, they undergo rest, and they do not perform any activity. They almost stop their metabolic reactions, and then these endospores can withstand dry conditions, and even they are resistant to temperatures. So either high temperature or very low temperature, and the thick wall which is surrounded, it becomes a wall which is very resistant for that cell, and also even when the cell is present in some poisonous chemical, there also the wall can resist. it can remain alive. This cell inside can remain alive without the reaction of the chemical on the cell wall. Means to such type of cell wall is synthesized by the bacterial cell, and this is called as endospore. It remains for a long period, and when favorable conditions arrive, after the unfavorable conditions they disappear, they are gone when favorable conditions appear. Now this cell will come out of the this envelope, means the parent cell. It will come out of this cell wall. The cell wall gets dissolved, and the cell inner comes out, and it will start this germination. Germination means fission. The outer cells get the thick wall gets ruptured, and then the cell will produce many cells by binary fission. So that is how the this protoplast will escape from the cell wall, and then it will produce new cells by the process of binary fission. So this is about the asexual reproduction of the bacteria. So bacterial cells, they reproduce by binary fission, and during favorable conditions, and during unfavorable they reproduce by these endospores. So endospore, strictly when we study, it is not reproduction, because actually reproduction means there should be increase in the number of the cells, isn't it? But here in endospore formation, the number of cells is not increased. Instead, the existing cell is survived. So the existing cell remains, and that is how endospore formation, it retains the generation of that particular species of that cell. But in fission, the number goes on increasing. For every 20 minutes, the number becomes double in number. Now, for example, if there is 1 million of bacteria in our environment, in the next 20 minutes it becomes 2 million, in the next 20 minutes 4 million, in the next 8 million. But however, this increase of bacteria is not seen in the environment, because of the conditions are not always favorable for fission. Suppose if there is favorable environment, definitely the number of the bacteria will go on increasing, and thus there will be heavy population of bacteria in a particular environment. So this is about the structure of bacterial cell and the asexual reproduction of the bacterial cell. So bacteria are unicellular prokaryotes. They are always unicellular. We never come across multicellular bacteria. But however, they appear to be multicellular when they are found in filamentous forms or in colonial forms. So though they are found in colonial and filamentous, which are covered by a mucilaginous envelope, but there is no any division of labor among the cells. All the cells, though they are adjacent to each other, they live independently, and they conduct their own metabolical process, and there is no any division of labor in the colony or in the filament. And that is about the bacterial structure, and also the asexual reproduction of bacteria.